Welcome. In this session, we'll discuss the anatomy of the neurosensory retina. In the previous session, we discussed the anatomy of the vitreous, and in the next session, we'll discuss the function of neurosensory retina. We'll discuss the anatomy of the retinal pigment epithelium and the choroid in a subsequent session. The neurosensory retina converts light signal into an electrical signal and is thickest at the foveal rim and thinnest at the foveola and the ora serrata. We'll discuss the fovea, the foveola and the ora serrata regions of the neurosensory retina when we discuss topography. The neurosensory retina contains three classes of cells: neurons, glia and vascular cells. Among the neurons we have rod and cone photoreceptors, bipolar cells, ganglion cells, horizontal cells and amacrine cells. Among the glia we have the Muller cells and also astroglia and microglia. Among the vascular cells we have endothelial cells and pericytes. We'll be discussing the neuronal and the glial cells in more detail in this session. The major route of information flow from the retina to the optic nerve is from the photoreceptors to the bipolar cells and from the bipolar cells to the ganglion cells. The horizontal and the amacrine cells are considered as interneurons which modulate this information flow. The anatomy of the retina can be described by its layers and by its topography. The layers of the retina are visible in a cross-section of the retina, in histological sections and also on optical coherence tomography or OCT. And the topography of the retina describes the surface features of the retina which can be distinguished when we examine the retina in phase during ophthalmoscopy. The relative positions of the layers of the retina can be described as proximal or inner, which are nearer to the vitreous, and distal or outer, which are nearer to the choroid. All the layers of the retina, as we will be describing, are found throughout the retina except at the ora serrata and at the fovea. The inner or the proximal layers of the retina are displaced peripherally at the fovea. The neurosensory retina and the retinal pigment epithelium constitute the retina. We will be discussing the retinal pigment epithelium in a subsequent session. From proximal to distal, the neurosensory retina constitutes of the internal limiting membrane, the nerve fiber layer, the ganglion cell layer, the inner plexiform layer, the inner nuclear layer, the outer plexiform layer, the outer nuclear layer, the external limiting membrane, the inner segments of the photoreceptors and the outer segments of the photoreceptors. The potential space between the neurosensory retina and the retinal pigment epithelium is called the subretinal space and is the plane of separation in retinal detachment. The external limiting membrane is said to form the roof of the subretinal space, and accordingly the inner and the outer segments of the photoreceptors lie in the subretinal space with interphotoreceptor binding matrix between them. The adhesion between the neurosensory retina and the retinal pigment epithelium occurs through pump function of the retinal pigment epithelium, the interphotoreceptor binding matrix, and outward pressure from the vitreous body. We will be describing the layers of the neurosensory retina from the distal side to the proximal side in the direction of flow of the visual information. And most distally in the neurosensory retina we have the photoreceptors. So to reach the photoreceptors, light rays have to travel through the entire thickness of the neurosensory retina. On being stimulated by light, the photoreceptors send signal to the bipolar and the horizontal cells. On stimulation by light, photoreceptors actually hyperpolarize, that is, they become electrochemically inactive, and this is very important to remember. Unstimulated photoreceptors secrete glutamate at their axonal endings, and upon stimulation by light, the secretion of glutamate from their axonal endings actually decrease. The parts of a photoreceptor cell are the outer segment, the cilium connecting the outer segment with the inner segment, the body containing the nucleus, the axon, and the synaptic terminal. There are two types of photoreceptors, the rods and the cones. The rods have the highest sensitivity in scotopic conditions, and they have a cylindrical outer segment. They constitute 95% of all photoreceptors in the retina and number around 100 million in a retina. They have the highest density 4 mm peripheral to the center of the fovea, and the density of the rods decrease further peripherally. The rods are not present in the fovea, the central area of 1.5 mm diameter of the retina, and this is very important to remember. The cones have the highest sensitivity in photopic conditions and provide high spatial acuity and color vision. They have tapering outer segments and they constitute only 5% of all photoreceptors in the retina, numbering 5 million in a retina. The cones have the highest density in the fovea, but still 90% of all cones remain outside the fovea. In the fovea, we have only the red and green cones, and the blue cones are absent in the foveal area. The outer segment of the photoreceptors is the location where photons are absorbed and phototransduction occurs. Phototransduction is the conversion of the photon signal to electrical energy, which we will be describing in detail in the next session. The outer segment contains membranous discs which contain the visual pigment. These discs are formed in the inner end of the outer segment by invagination of the plasma membrane, and they receive their protein and lipid components from the inner segment. Docosahexaenoic acid is the major lipid in the rod discs, which needs to be obtained from diet. And the proteins include the opsins or the visual pigments and other proteins such as ABCA4 in rod discs. Mutations in ABCA4 has been implicated in Stargardt's disease, retinitis pigmentosa, and age-related macular degeneration. Distally, the outer segment opposes to the apical region of the RPE cells, but without any intercellular connections between them. And proximally, the outer segment connects with the inner segment through the cilium. The rod outer segment is cylindrical in shape, and the mature discs in the rod outer segment are discontinuous from the plasma membrane of the outer segment as shown here in this diagram. These discs are shed from the outer end of the outer segment, phagocytosed by the retinal pigment epithelium and degraded. And shedding of discs from rod outer segments occur at light onset when rods are no longer required for visual function. The major protein in the rod membranes is rod opsin, which is coupled with the chromophore 11 cis retinal, and this molecule together is called rhodopsin. There are about 1000 discs in a rod outer segment, and each disc contains approximately 1 million rhodopsin molecules. Additional proteins which are present in rod membranes include peripherin and ROM1, which are found in both rods and cones, and ABCA4, which we have just mentioned, which are found in rods only. Peripherin and ROM1 are found near the rim of the membranous discs and help in maintaining the curvature of these membranous discs. And ABCA4 is a transporter molecule which transfers all transretinal across the disc membrane. The cone outer segment is approximately half the length of the rod outer segment, wider at its inner end and tapers towards its outer end. And at least several, if not all, mature discs in the cone outer segment remain connected to the plasma membrane of the outer segment of the cone, increasing the area for exchange of proteins and ions. And this is another difference between the cone outer segment and the rod outer segment apart from their shape. The cone outer segment discs are recycled by Muller cells and occurs at light offset when cones are no longer required for visual function. The cilium connects the outer segment to the inner segment and is non-motile in character without central doublet of fibers. They originate from basal bodies in the inner segment and extends through the entire outer segment. The inner segment of photoreceptors consists of two parts: the ellipsoid containing mitochondria located adjacent to the outer segment, and the myoid containing endoplasmic reticulum adjacent to the body containing the nucleus. The next proximal portion of the photoreceptors is the body containing the nucleus, which is located in the outer nuclear layer. The next proximal portion of the photoreceptors is the body of the photoreceptor containing the nucleus, which is located in the outer nuclear layer. The outer plexiform layer is formed by synaptic connections between photoreceptors, bipolar cells, and horizontal cells. The rods connect with a single type of bipolar cells called the rod bipolar cells, while the cones connect with several types of bipolar cells. The rod terminals called spherules connect with two horizontal cells and two rod bipolar cells, whereas cone terminals or pedicles connect with several horizontal and bipolar cells. The cone terminals also receive connections from rods and other cones. A single cone bipolar cell connects with fewer cones as compared to the number of rods a single rod bipolar cell connects with. As we have mentioned, the synaptic terminals of photoreceptors continuously secrete glutamate in darkness, and on exposure to light, the photoreceptors become deactivated and the secretion of glutamate decreases. The inner nuclear layer contains cell bodies of four types of cells: the bipolar cells, the horizontal cells, the amacrine cells, and the Muller cells. The bipolar cells are said to be the first neurons of the visual pathway and they synapse with the ganglion cells. They have glutamate as neurotransmitter at their axonal endings, and there are around 13 types of cone bipolar cells, but only one type of rod bipolar cell, as we have just mentioned. There is a major functional division of cone bipolar cells which is preserved up to the higher visual functions. The bipolar cells can be either on bipolar cell or off bipolar cell. The on bipolar cells are optimized to respond to increases in intensity of light stimulus, and they have inhibitory glutamate receptors in their dendrite terminals. So what happens is, on receiving a light signal, the photoreceptor deactivates, decreasing secretion of glutamate in their synaptic terminals, and this releases the inhibition on inhibitory glutamate receptors on the on bipolar cells. So the on bipolar cells are stimulated by increases in intensity of light. On the other hand, off bipolar cells are optimized to respond to decreases in intensity, and they have excitatory glutamate receptors in their dendrites. So with decrease in light intensity, the photoreceptor is activated, secrete glutamate from its synaptic terminals, which in turn stimulates excitatory glutamate receptors on the off bipolar cells, and thus the off bipolar cells are stimulated when there is decrease in intensity of light. The midget cone bipolar cell is a special type of bipolar cell which is the predominant type of bipolar cell in the central retina. One off midget cone bipolar cell and one on midget cone bipolar cell synapse with each cone in the central retina, and the axon of each midget cone bipolar cell synapse with a single midget ganglion cell. And because of this 1:1 transmission of information from the cone to the midget bipolar and then to the ganglion cell, the central retina is responsible for high spatial acuity. As we have mentioned, there is only one type of rod bipolar cell, and each rod bipolar cell is said to connect with approximately 80 to 100 rods, resulting in high sensitivity to relatively few photons. The rod bipolar cells are all on in nature, having inhibitory glutamate receptors in their dendrites. There are no separate ganglion cells for rods, which is important to remember. And most rod bipolar cells synapse with the A2 amacrine cells, which connect with on and off cone bipolar cell. And another alternate rod pathway is said to exist as gap junction between rods and cones. So rods don't have their own ganglion cells and take the help of cone bipolar cells and cones to transmit their information. Horizontal cells are interneurons located in the outer aspect of the inner nuclear layer and communicate with photoreceptors. They have a large receptive field from multiple photoreceptors and send negative feedback signals to the photoreceptors, and they are said to play a role in enhancing response to low contrast targets and also increase the threshold of flicker fusion in rapidly alternating targets. Flicker fusion is the frequency of alternation of a rapidly alternating target when the alternation is no longer perceived and the target is seen as a continuous stimulus. The amacrine cells are located in the inner aspect of the inner nuclear layer, and they are involved in feedback inhibition of the bipolar cell and feed-forward inhibition of the ganglion cell. Several types of amacrine cells exist. The A2 amacrine cells, as we have seen, play a role in maximum scotopic acuity, the S1 and S2 amacrine cell provide negative feedback on rod bipolar cells. The dopaminergic amacrine cell play a role in light adaptation, which we will discuss in the next session. And the starburst or the cholinergic amacrine cells are involved in directional sensitivity to moving stimulus and have connections to directionally sensitive ganglion cells. The Muller cells are the predominant glial cells of the retina, and there are approximately 4 to 5 million Muller cells regularly arranged in the neurosensory retina. So there are approximately the same number of Muller cells as cones. The cell body of the Muller cells lies in the inner nuclear layer with two long processes extending up to the internal limiting membrane and the external limiting membrane. So the Muller cells extend through almost the entire thickness of the retina. Lateral processes arise from the long processes of the Muller cells, and they enclose synaptic junctions and cell bodies of neurons in the neurosensory retina. The predominant role of the Muller cells is to maintain homeostasis of the extraneuronal environment. They convey nutrients and waste products between the neurons and the blood vessels, the subretinal space, and the vitreous cavity. They also play an important role in the maintenance of inner blood-retina barrier, recycling of neurotransmitters, and recycling of cone outer segments and their visual pigments. Muller cells also play a role in immunological reactions and repair following injury. Here we'll discuss the glial cells of the retina, and the glial cells of the retina can be classified as microglia and macroglia. The microglia are the blood-derived resident mononuclear phagocytes present mostly in the ganglion cell layer, the nerve fiber layer, and the plexiform layers, and they play a role in host defense, immunological reactions, synapse remodeling, and tissue repair. Among the macroglia, we have the Muller cells and the astrocytes, and the astrocytes are immigrants from brain and reside in the ganglion cell layer and the nerve fiber layer. Astrocytes take part in maintaining homeostasis and repair of ganglion cells following injury, and they play a role in limiting extent of neuronal injury by taking up glutamate, producing neurotrophic factors, and restoring blood-retina barrier. So the Muller cells are the only glial cells which are present throughout the retina, whereas the microglia and the astrocytes are present mostly in the ganglion cell layer and the nerve fiber layer on the inner aspect of the retina. The inner plexiform layer is formed by synaptic connections between bipolar cells, the ganglion cells, and the amacrine cells. The ganglion cells are said to be the second neuron of the visual pathway, and there are approximately 1.5 million ganglion cells in each retina. There are more than 40 types of ganglion cells, and the ganglion cells receive input from the bipolar cells and the amacrine cells. As we have discussed, the on-ganglion cells receive input from on-bipolar cells, and the off-ganglion cells receive input from the off-bipolar cells, and this on-off separation is retained till the higher visual centers. The ganglion cell processes constitute the nerve fiber layer and project predominantly in the lateral geniculate nucleus. The vast majority of ganglion cells secrete glutamate as a neurotransmitter from their synaptic endings in the lateral geniculate nucleus. Here we should note that not all ganglion cells terminate in the lateral geniculate nucleus, as we have discussed in the neuro-ophthalmology section. Ganglion cells can also be functionally categorized as tonic or phasic. Tonic ganglion cells respond throughout the duration of the stimulus, and phasic ganglion cells respond only at the beginning and at the end of the duration of the stimulus. The midget ganglion cells which receive stimulus from cones are tonic, and as we have mentioned, there are no separate ganglion cells for rods. The parasol ganglion cells are phasic and are involved in motion detection. The midget ganglion cells, both on and off types, constitute the predominant ganglion cell in the central retina, and as we have mentioned, they connect with on and off types of midget cone bipolar cells in a 1:1 ratio, and so are responsible for high spatial acuity and color vision. And the midget ganglion cells project to the parvocellular layer of the lateral geniculate nucleus. The parasol ganglion cells, also called M cells, are involved in motion detection, contrast sensitivity, and depth perception, and they project to the magnocellular layers of the lateral geniculate nucleus. The bistratified ganglion cell provides blue-yellow color function, whereas the midget ganglion cell provides red-green color function. The bistratified ganglion cells receive signal from blue cone cells through blue bipolar cells, and the bistratified ganglion cells send signal to the coniocellular layer of the lateral geniculate nucleus. Then there is the directionally sensitive ganglion cell, which responds maximally when a linear stimulus moves in one direction and does not respond when the stimulus moves in the other direction. And as we have mentioned, they receive input from the starburst amacrine cells. The intrinsically photosensitive retinal ganglion cells, or the IPRGCs, can respond to light in the complete absence of rods and cones. They contain a visual pigment called melanopsin, and they may play a role in pupillary response and circadian rhythm, and so through this pathway pupillary responses and circadian rhythm may be present in a patient with complete destruction of photoreceptors. The nerve fiber layer is constituted by the axons of the ganglion cells traveling towards the optic nerve. And we have discussed the nerve fiber layer in further detail in neuro-ophthalmology and glaucoma sections. Then there are the membranes in the retina, which are not true membranes in the sense that they do not bleed when they are peeled. They can be detected histologically and on optical coherence tomography. The internal limiting membrane covers the inner surface of the retina and is formed by the footplates of the Muller cells along with their basement membrane. This basement membrane is smooth on the vitreal side and follows the contour of the Muller cells on the retinal side. The middle limiting membrane is located in the inner third of the outer plexiform layer and is formed by synaptic connections and cellular adhesions between the photoreceptors and the bipolar cells. Retinal blood vessels do not usually extend distally beyond the middle limiting membrane, and so the photoreceptors are supplied by the choroidal circulation, whereas the remaining inner layers of the retina are supplied by the retinal circulation. The external limiting membrane, or the ELM, is formed by cellular adhesions between the photoreceptors and the outer ends of the Muller cells. The external limiting membrane lies at the level of the junction between the body and the inner segment of the photoreceptors. The external limiting membrane is said to form the roof of the subretinal space, and the external limiting membrane is highly permeable, allowing passage of oxygen and macromolecules between the choroid and the outer retina. The anterior margin of the retina is formed by the ora serrata. We'll describe the surface features of the ora serrata later when we discuss topography of the retina. At the ora serrata, the internal limiting membrane is continuous with the basement membrane of the non-pigmented epithelium of the ciliary body. The neural retina is continuous with the non-pigmented epithelium of the ciliary body. The external limiting membrane and the apical junctions of the retinal pigment epithelium are continuous with the apical junction system between the non-pigmented and the pigmented epithelium, and they thus form the anterior border of the subretinal space. It is called an apical junctional system because the apical portion of the cells of the non-pigmented ciliary epithelium face the apical portion of the cells of the pigmented ciliary epithelium. The retinal pigment epithelium is continuous with the pigmented epithelium of the ciliary body, and the Bruch's membrane is continuous with the basement membrane of the pigmented epithelium of the ciliary body. The posterior margin of the retina lies at the optic nerve head. Here the internal limiting membrane is continuous with the basement membrane of the Elschnig and the central glial meniscus of Kundt on the anterior surface of the optic nerve head. The external limiting membrane is continuous with the apical junctional girdles of the retinal pigment epithelium at the intermediary border tissue of Kundt, which separates the axons of the optic nerve head from the outer retina and the retinal pigment epithelium. Posterior to the intermediary border tissue of Kundt lies the intermediary border tissue of Elschnig, which separates the axons of the optic nerve head from the choroid. The topography of the retina describes the surface features and regions of the retina, and we also need to correlate it with the characteristic features of the layers of the retina in its different regions. The macula is also known as the area centralis, and it is an area 5.5 mm in diameter bordered by the temporal vascular arcades and the optic disc. And the macula is characterized by two or more layers of ganglion cells in the ganglion cell layer. A high concentration of oxygenated carotenoids, lutein and zeaxanthin are present in the outer plexiform layer of the macula, giving it a yellow color, particularly when the macula gets detached and in cadaveric eyes. The fovea centralis is a central area of 1500 microns diameter, which is approximately the size of the optic disc, and so is said to be 1 disc diameter or 1 dd in size, which is very important to remember. The retina is thickest at the margin of the fovea, and here the thickness of the retina is around 550 microns. The fovea contains only cones as photoreceptors, which is also very important to remember. So the fovea is responsible for high spatial acuity and color vision functions. In the fovea, only red and green cones are present, blue cones are absent in the fovea. The fovea slopes 22 degrees centrally and towards the retinal pigment epithelium. What is important is that there is a centripetal or towards the center displacement of the photoreceptors, and centrifugal or away from the center displacement of the remaining cells, the ganglion, bipolar and Muller cells, during foveal maturation, which takes place between 3 months before and 3 months after term. As a result of this displacement, the fibers in the outer plexiform layer are oriented obliquely, more radial than vertical, as they run from the photoreceptors in the outer nuclear layer to the bipolar cells in the inner nuclear layer. The outer plexiform layer is thick in the fovea because of larger density of the photoreceptors, and this layer of the retina in the fovea is also called the Henle's layer. Because of this displacement in the center of the fovea, there is minimum scattering of light in the center of the fovea before the light reaches the photoreceptors. And also because of this displacement, the bipolar and the ganglion cell layers are multilayered. The floor of the fovea is called the foveola, and it is 350 microns in diameter and is located 4 mm temporal to or 2.5 disc diameters temporal to and 0.8 mm inferior to the center of the optic disc, and this relation is very important to remember. As we have just described, the Henle's layer or the outer plexiform layer in this region is radially oriented and is almost parallel to the inner retinal surface in this region, resulting in petaloid or star-shaped patterns when the extracellular spaces of this layer get filled with fluid or exudate. In the foveola, the cell bodies of the photoreceptors in the outer nuclear layer bow anteriorly, and this is called the fovea externa. The outer segments of the photoreceptors in the foveola are oriented parallel to the visual axis, but the outer segments of photoreceptors in the peripheral retina are tilted towards the entrance pupil. The internal limiting membrane becomes very thin in the foveola. The umbo is the depression in the center of the foveola, which is 150 to 200 microns in diameter, and it represents the center of the fovea and the macula and corresponds to the foveal light reflex. The only cells present in the umbo are red and green cones and Muller cells. The foveal avascular zone is a feature of the center of the macula on fluorescent angiography and optical coherence tomography angiography. This photograph shows an optical coherence tomography angiogram showing the foveal avascular zone. The foveal avascular zone is centered on the foveal center and is approximately 250 to 600 microns in diameter, and thus corresponds more or less with the foveola. It is entirely perfused by choriocapillaries and does not receive any blood supply from the retinal circulation, and thus can be severely affected if the retina gets detached in this region. The perifovea is an annular area of 0.5 mm width surrounding the fovea. Here the retina is thickest because the ganglion cell layer, the inner nuclear layer and the outer plexiform layer are the thickest here, because of the centrifugal displacement which we have discussed. The Parafovea is an annular area of 1.5 millimeters width surrounding the Perifovea, and the outer margin of the Parafovea delimits the macula. The retina peripheral to the macula contains a single layer of ganglion cells in the ganglion cell layer, and the periphery is divided into near periphery, the middle periphery, the far periphery, and the extreme periphery or the ora serrata. The near peripheral retina is a 1.5 millimeter ring peripheral to the temporal major vascular arcades. The middle peripheral retina or the equatorial retina is a 3 millimeter ring peripheral to the near peripheral retina. The far peripheral retina is anterior to the equatorial retina up to the ora serrata. And the extreme peripheral retina is called the ora serrata, which is the junction between the pars plana and the retina. The ora serrata is located on average 5.75 millimeters on the nasal quadrant and 6.5 millimeters on the temporal quadrant from the Schwalbe's line in the region of the spiral of Tillow. The spiral of Tillow is the line joining the insertion of the rectus muscles. So the ora serrata is nearer to the limbus in the nasal quadrant as compared to the temporal quadrant, and this is an important relation to remember. The ora serrata is more serrated in the nasal quadrant, and retinal blood vessels end before reaching the ora serrata. The ora serrata lies in the watershed region between the vascular supplies to the anterior and posterior segments of the eye. Dentate processes are extensions of retinal tissues onto the pars plana, and they are more prominent nasally, giving the more serrated appearance of the ora serrata in the nasal quadrant. Oral bays are the extensions of the pars plana onto the peripheral retina, and portions of the oral bays may be enclosed by dentate processes, which are called enclosed oral bays. Meridional folds are radially oriented folds of retinal tissue extending onto the pars plana, and retinal tears often occur posterior to the meridional folds. A meridional fold may be aligned with an enlarged ciliary process, and these two together are called meridional complex. To recap the salient points, retina is the light-sensitive tissue which converts light energy to electrical energy. It consists of a neurosensory layer and a retinal pigment epithelium separated by a potential space called the subretinal space. The layers of the neurosensory retina from the choroidal aspect to the vitreal aspect are as follows. The layer of rods and cones contain the outer and inner segments of the photoreceptors, and there are two types of photoreceptors: rods mediating scotopic vision and cones mediating photopic vision, high spatial visual acuity and color vision. The outer segments of the rods are cylindrical and that of the cones are conical. The outer segment contain membranous discs containing the light-sensitive visual pigments. The discs are attached to the plasma membrane of the outer segment in cones, but not in rods. The outer and inner segments are connected by cilium, which transfers proteins and lipids from the inner segment to the outer segment. The inner segment contains mitochondria distally in the ellipsoid region and endoplasmic reticulum proximally in the myoid region. The external limiting membrane lies at the level between the inner segments and the cell bodies of the photoreceptors. The outer nuclear layer contains the bodies of the photoreceptors with their nuclei. The outer plexiform layer contains the synapses between photoreceptors, bipolar cells, and horizontal cells. The middle limiting membrane lies in the inner third of the outer plexiform layer. The inner nuclear layer contains the cell bodies of the bipolar cells, horizontal cells, Amacrine cells, and Muller cells. The bipolar cells carry the primary visual information from the photoreceptors to the ganglion cells. There are several types of cone bipolar cells, both on and off in nature, but only one type of rod bipolar cell, which is only on in nature. Midget cone bipolar cells are the predominant type of bipolar cells in the central retina, providing 1:1:1 connection between photoreceptors, bipolar cells, and ganglion cells, thus allowing high spatial acuity. Horizontal cells are located in the outer aspect of the inner nuclear layer and synapse with a large number of photoreceptors and provide negative feedback to photoreceptors. Amacrine cells are located in the inner aspect of the inner nuclear layer and synapse with bipolar and ganglion cells. Muller cells are glial cells which span the entire width of retina from the external limiting membrane to the internal limiting membrane. The inner plexiform layer contains synapses between bipolar cells, amacrine cells, and ganglion cells. Cone bipolar cells have their own ganglion cells, but rod bipolar cells do not. Rod bipolar cells send their visual information to cone bipolar cells through A2 amacrine cells. The ganglion cell layer contains ganglion cells. Midget ganglion cells are involved in high spatial acuity and red-green color vision. Bistratified ganglion cells are involved in blue color vision. Parasol ganglion cells are involved in motion detection, and intrinsically photosensitive retinal ganglion cells are involved in pupillary response and circadian rhythm even in the complete absence of photoreceptors. The nerve fiber layer contains the axons of the ganglion cells on their way to the optic nerve. The internal limiting membrane is the inner lining of the retina formed by the footplates of the Muller cells and their basement membrane. Topographically, the retina has a central region and a peripheral region. In the central region, the macula is the 5.5 mm diameter region of the retina between the optic disc and the temporal vascular arcades. The ganglion cell layer has more than one layer of ganglion cells in this region. The carotenoids lutein and zeaxanthin are present in the outer plexiform layer in this region. The fovea is the central 1.5 mm diameter area within the macula. It is approximately 1 disc diameter in size. It contains only cones as photoreceptors. The fovea slopes centrally towards the retinal pigment epithelium. This shape is formed in the perinatal period by the centripetal displacement of the photoreceptors and the centrifugal displacement of all other cells in the retina. The margin of the fovea is the thickest portion of the retina. The foveola is the 350 micron floor in the center of the fovea. It is located 2.5 disc diameters temporal to and 0.8 mm inferior to the center of the optic disc. Because of the displacement during foveal maturation, the outer plexiform layer called the Henle's layer in this region is oriented radially instead of vertically, lying almost parallel to the inner surface of the retina. This gives rise to the petaloid appearance clinically to extracellular accumulation of fluid and exudates in the Henle's layer. The foveal avascular zone on fluorescent angiography and optical coherence tomography angiography approximately corresponds to the foveola. The umbo is the central 150 to 250 micron depression in the foveola, where the only cells present are the red and green cone photoreceptors and Muller cells. The perifovea is a 0.5 mm ring around the fovea. The parafovea is a 1.5 mm ring around the perifovea, delimiting the macula. Peripheral to the macula lies sequentially the near periphery, which is a 1.5 mm ring around the macula, the equator or mid-peripheral retina, which is a 3 mm ring peripheral to the near periphery, and far periphery extending from the equator to the ora serrata. The ora serrata is the extreme periphery of the retina and is the junction between the pars plana and the retina, which is closer to the limbus and more serrated in the nasal quadrant than in the temporal quadrant. Dentate processes are projections of the retina onto the pars plana. Oral bays are extensions of the pars plana onto the retina, some of which can be enclosed by adjacent dentate processes. Meridional folds are folds of peripheral retina extending into the pars plana along dentate processes. When meridional folds align with enlarged ciliary processes, it is called a meridional complex. Retinal tears are likely to occur at the posterior margins of meridional folds. Thank you for listening.